Welcome to the Genomics Revolution, the podcast exploring how we're making the invisible visible to deliver real world health benefits. I'm Sian Jarvis, and in this series, we'll uncover the potential of genomics to help people live longer, healthier lives by transforming health and reshaping drug discovery. In this first episode, I'm joined by one of the leading experts in the field, Professor Sir Peter Donnelly, founder and chief executive of Genomics Ltd and emeritus professor of statistical science at the University of Oxford. He's played a leading role in national and international genomics projects, including the HapMap project and with the Wellcome Trust Case Control Consortium. Welcome, Peter. I introduce this series as the Genomics Revolution. I guess for somebody who's given their whole life to the study of genomics, it probably doesn't seem much like a revolution, more an evolution. Which do you think it is?
I think it really was a revolution. From about 2005 or 2006 onwards, there was a complete transformation in our understanding of the way in which little bits of our DNA affected people's risk of all of the common diseases. Before that time, across many, many common human diseases, we knew only a small number of examples of how genetics affected people's risk. Now we know tens of thousands of examples of places in our DNA which either affects how likely someone is to develop a disease, or it affects their levels of something we can measure, whether that's blood pressure or cholesterol and so on. So absolutely, over that period of time, it was a revolution. We've been trying for 20 years to understand these problems. Suddenly we had a good way of tackling them, and there were extraordinary results over that period of 5 to 10 years, which has continued, and indeed laid the foundations for what we're able to do now.
And what are we able to do now? What would you how would you describe the impact of genomics? I guess both in healthcare and also in drug discovery.
Well, that's why we founded the company more than 10 years ago, to take advantage of those scientific discoveries and to put the pieces in place which would allow us to make a difference to people's lives. Part of that is about data and building the right data resource, and we've got an unparalleled resource inside the company. Another part is about the statistical and machine learning algorithms, which allow us to analyze that at scale and develop the insights from. And we use that in two different ways within genomics. One part of that is understanding risk for individuals. So measuring for each person what their level of risk is for each of the common diseases, for heart disease, for diabetes, for breast cancer if they're a woman, or prostate cancer if they're a man. And we can now get much, much more personalized and precise measures of risk for each of us.
Mm.
Some of us will be at very high risk for some of these diseases and we'll have no idea about them. So that side of the business is about getting much better estimates of risks when people are still healthy, in order to give them the information that'll help them make the right choices in their life, but critically also to give their doctors and their health systems the information that allow them to do stuff health systems are already doing, to get the right people into the right prevention programs, the right screening programs, and the right treatment programs at the right time.
Well, let's come back to that. But if we look at drug discovery in particular, I mean, one of the things I do know is that 95%, it's a staggering statistic, 95% of drugs fail during that development process. I mean, why is it so hard? Why is it so difficult? Why does it take so long to develop drugs? And how can this help?
It's a great question, and I think genomics can help a lot. But let me go back to the first part of your question. Why is it so hard? I think the reason is that fundamentally, when we give someone a drug, we're trying to change some aspect of human biology. We're trying to make it slightly different to improve outcomes for the disease in question. Normally, when we do science, we have a very robust scientific method. You have a hypothesis, you do an experiment, you learn from the experiment, and you update your hypothesis. The problem with human biology is, for very good reasons, we can't do experiments on humans. So we can't study directly the very thing we're trying to change, which is human biology. We have to use surrogates for it. And so people doing drug discovery do that in different ways. They sometimes use animal models that they hope will replicate factors of humans, or they use cells from humans in a dish, again, where they hope that will recapitulate the full organism's biology. And what we're learning is, while these tools can be helpful, they can often be fallible. And that's the key problem, the fact that we can't do experiments and we can't do biology directly in humans. Where genetics helps is it gives us a way of understanding human biology much more directly. So when someone thinks of developing a new drug therapy at the moment, they will have in mind a target. So that's a protein typically that they'll want to change. They'll want to stop it from working. That would be called inhibiting the protein. And they hope that'll be useful for and helpful for the disease in question. Let's say, for this example, diabetes. At the moment, to know whether they're right, they have to do two different things, both of which are expensive and time consuming. The first one is, if they have that hypothesis, they have to develop a molecule, physical molecule, that they get into the right part of the body and it interacts with the protein to stop it doing what it would normally do. That's the first part. That's already pretty hard. And then the second part is to see whether that change is actually helpful for the disease. And that's what we do in clinical trials. And actually, to go back to your original number, the fact that only 5% of drugs succeed in trials, sometimes
So, from what you've just described, I begin to see why drug development takes so long and why it costs what £2.6 billion to create a drug. So, how can genomics help in that process?
well, the key insight is that when you give someone a drug, you're trying to change some aspect of their biology. For example, stopping a protein from doing what we normally do. We all carry genetic variants. Some letters in our DNA that one of us will have and another person won't have. And some of those variants change our biology as well. So the simple idea is, if we can find people who have genetic variants that change the bit of biology around what your drug target's proposing, then we can use those genetic changes to gain insights in humans about what happens if you make that change. So, for example, if you want to have a drug that stops a protein from working, we can probably find genetic variants which mean that some people just make a bit less of that protein than others. For those people, it's like they've been on a weak version of your proposed drug all their life. So we can just look in the data and see, do they get less diabetes, in our example, the disease you're trying to treat? If they do, that's really helpful. It's telling you that, actually, I know from changes in humans that affecting that protein is helpful for diabetes. If they don't, that can be worrying. But we can go beyond that, as well as learning about the effects on the disease you're interested in. You can also see whether that particular change has any other consequences. Are there people who it's like they've been on a weak version of your drug all their life? Maybe they get more prostate cancer if they're men. So that's giving you information that change that we're proposing can also cause side effects. So you can not only learn whether the drug is helpful for the disease you're interested in directly through human biology, you can learn what other consequences and what side effects there might be.
Peter, I mean, that's great in theory, but what's the evidence that it actually makes any difference?
It's a really good question. There's now quite a lot of evidence. So a number of different studies have been undertaken where they look at drug targets which have that human genetic support and ask whether they're more likely to get through the various stages of a clinical trial and to succeed overall. And what that evidence shows, and there are multiple different papers now, is that drug targets with human genetic support are at least twice as likely to succeed in clinical trials. And that doubling of the success probability is huge in the context of pharmaceutical development. So
I mean,
that's how genetics can help and does help. We believe that if you use it in sophisticated ways, you can probably increase the success probability by even more than that.
that's incredible. So that 95% suddenly starts to come right down.
Correct.
And how else can you use genomics in the kind of actually maybe identifying some of the participants who come forward for the clinical trials? Is that one?
Yes. Yes, the way I like to think about it is that we can help upstream in drug development in helping companies, whether they're biotechs or established pharmaceutical companies, help them choose the right targets for potential new medicines. And then once a company has a target, we can use genetics to help them choose the patients who will most benefit from that. And that's a newer area, but I think one which is really exciting. There's now evidence in a couple of different areas where selecting people on the basis of their genetic risk for the disease identifies individuals who are more likely to respond. Sometimes that's useful for the initial drug, but it can also be really useful when a drug company has a successful drug for one disease and they're thinking of what's called an indication expansion. Can they find a related condition where the same drug might be helpful? Genetics can be and has been really, really powerful there as well.
And you're working with a number of life science companies already. I mean, are there any particular examples that you're able to talk about?
Yes, a lot of it's confidential, but for example, we have a long-standing partnership with Vertex Pharmaceuticals. Vertex, I think, are one of the leaders in the industry at really putting effort into finding the right drug targets before they develop programs. And we partner with them to either help them find new drug targets using our data, our tools, and our scientists, or to validate, to tell them what human genetics says about a drug target they may already be thinking of. That partnership has been really successful. It's been renewed twice already, most recently for another 3 years, because of how positive Vertex are about the impact we're having. In another collaboration which has been announced, we've worked with GSK to look at the impact of genetics and genetic risk in identifying the right patients to most benefit from particular therapeutic drugs.
So we talked there about how genomics can help drug discovery. We also know it helps in health, and I know with your polygenic risk scores that you have developed, we can move healthcare to being much more predictive and proactive. How can that happen? How can that be used, and what diseases can we spot early?
I think there's a lot of potential for us to get much better at identifying risk on an individual basis. And when we have better estimates of risk for individuals across many of the common diseases, then we have the potential, first of all, to empower the individuals. I like to talk about making individuals co-pilots in their health journeys rather than just passengers. They can know what are the things they can do to reduce the particular risks they have. But then we can help health systems. We can help doctors make better recommendations to patients. So, for example, in cardiovascular disease, doctors currently estimate patients' risk when they're middle-aged. They do that with algorithms that combine age and sex and blood pressure and cholesterol level and so on. Genetics is a really important risk factor there. We don't currently measure it. For a man who's between 40 and 55, genetics captures as much risk by itself as does the current risk tool. So it can add massively to the ability of doctors to give the right advice to the right patients, and it changes the conversation with the patients.
Peter, you've talked a lot there about some of the studies, some of the evidence that you have about the ability of genomics to supercharge drug discovery. Why is there so much hesitation around the adoption of genomics? Why we've obviously got some people who are out there as first movers, but there are a lot of people who still need to catch up. I mean, that sounds like a real paradigm shift. It does sound like a revolution, the industrial revolution, but this is the digital revolution. How close are we to enabling that and allowing it to happen in a quite straightforward fashion, or does it require massive investment?
I think it's about thinking, and some companies have done this, about thinking how to have this human biology and evidence in human biology right at the center of a process for finding drug targets. As I said, often what's done now with companies realizing the genetics can be helpful is they do what they're doing anyway. They get to a stage with the target, and then they metaphorically, or in some cases literally, send a memo off to their human genetics colleagues and say, what can you tell me about this target for this disease? They wait patiently for a while because their human genetics colleagues are invariably stretched in answering lots of questions. They get answers back, which may or may not answer the direct question they want and may not be easy for them to interpret. And what we want to do is to empower the individual researchers so that they can have access to that information much more directly and in a way which fits in much better with the processes that they're doing anyway.
Interesting. So that's life sciences, looking at pharma drug discovery. Tell me a bit about health and how we can use genomics to improve health outcomes.
One way I like to characterize it is the following. Until a few years ago, if we had the entire DNA sequence on someone who was 40 and healthy, we'd learn something medically actionable in only about 1, maybe 2% of cases. And that's because genetics has played into medicine by what's called genomic medicine around rare diseases, around conditions in which a single change in our DNA has a very big impact. Often it'll stop a gene from working, it stops a protein being produced, and that can have serious consequences. So examples like cystic fibrosis or Huntington's disease,
Mhm.
muscular dystrophy, example of those kinds
Is
of
this where the
diseases.
BRCA gene fits in?
Yes.
Yeah.
Yep. That's an example of a single change that has
Yeah.
a big impact on a woman's risk for breast cancer. Thankfully, those mutations are very rare, the conditions are rare, and actually they're even collectively rare. And also many of them have impact very early in people's lives, often in childhood. So that if you get to 40 and you're healthy, there's probably not much left to find in your DNA of that kind, those individual changes that have massive impact. What's changed is that now, if I've got genetic information on a healthy 40-year-old, I'll learn something medically actionable in about 70% of cases. So that change from 1% to 70% is because we go from genetics focusing on rare diseases to being able to use it to understand the risk for all the common diseases, heart disease, diabetes, breast and prostate cancer, and so on. The conditions which cause most sickness in our populations, most premature death, and use most of the healthcare budgets.
And how reliable is that information? Is this an absolute predictor, or is this sort of, I guess, more of a risk stratification tool?
That's a really good question. It's a risk stratification tool. It's about understanding better what the risk is for each of us across a number of diseases. Health systems do a lot of things currently based on risk, and they do their best to estimate patients' risk, whether to advise statins for cardiovascular disease, what age to start screening for breast cancer, and so on. We could do that much, much better. Each of those opportunities will be much more effective if we have a better understanding of risk. There'll be people at high risk currently for heart disease who will be invisible to the NHS because they don't yet have high enough cholesterol or high enough blood pressure to be put on statins. But actually, we know that whatever their current blood pressure and cholesterol level, in many cases, reducing that will be helpful for their outcomes. So we can start to do that earlier when we understand risk, and we can warn the individuals of that particular risk and the things they can do about it. For screening programs in the UK, for example, all women get offered mammograms when they turn 50. Now we know, because of their genetics, some women will be at the same level of risk as a typical 50-year-old in their late 30s. If we knew that, it's bonkers to say, well, you're at high risk, but we're not going to start screening you until you turn 50 because that's the way we do it. Other women are at very low risk at age 50. So if we had the ability to understand risk and then direct the screening programs based on risk, who's at the highest risk now, not is someone 50 or not 50, we're using those programs more effectively. Each mammogram that a health system spends money on will be more effective because it's targeted at people at higher risk. for each colonoscopy and so on.
And you have, again, evidence that underpins this research. Why hasn't the NHS, why haven't health systems adopted this at scale?
There's growing evidence that this improved risk prediction can make a difference for individual patients and for health systems. Both evidence through analysing large biobanks to see how knowing the genetic risk might have made a difference, but also through trials. We undertook a trial in the National Health Service with primary care practitioners, GPs as we call them in England, to add the genetic component of risk to what they're already doing in estimating risk for heart disease and stroke and cardiovascular disease. It was really successful. It showed that this fitted in with their workflows. The doctors were very positive about it. 98.5% of the patients found it helpful, and it changed the management of the patients in 13% of cases. So that piece of evidence is there. In the UK, there's a very large program called Our Future Health that's aimed to recruit up to 5 million people. They'll be given estimates of their risk for disease based on their genetics. So in the UK, in a few years' time, there'll be millions of people who understand their genetic risk through the tools we're developing. Just today, it was announced that one of the largest private providers in the UK is going to roll out this technology, our technology, in their health assessments. So it's starting to gain traction. But health systems are conservative. It has many advantages. And also, at the moment, it's particularly true in the UK of the NHS, they've got massive challenges with delivering care, long waiting lists, issues that they have to deal with, which are very front of mind. So doing stuff that's beneficial for the longer term tends to get put on the back burner and is less of a focus.
And I guess I mean, there are still challenges ahead, obviously, with any kind of new science and trying to operationalize it in a health system. But one of the aspects of, or one of the challenges is actually getting individuals to connect to their health. And I wonder, from your experience, whether you've seen that this enables that to happen, perhaps in a more sort of a more impactful way than has done before. You know, if you actually have some information about your risk of a particular disease, you can have a conversation which is somehow different.
Yes, it's early days yet in terms of that kind of evidence, but there are a number of encouraging signs. So some studies have found that when people know about their genetic risk of the kind I'm talking about, that changes their behavior and their engagement with doctors to take the right medicines. The anecdotal evidence from the trial we ran was that armed with this extra information, GPs had much more meaningful conversations with their patients, both about lifestyle changes they could make, but also about, for example, whether to go on statins.
And we've talked about the common diseases. Where do you think this could go next? Alzheimer's, you haven't mentioned. Is that a disease that we could begin to have much clearer picture around in terms of prediction?
We focused primarily on diseases where health systems already have programs for people who are at high risk. That's not yet the case for Alzheimer's disease, but that might change relatively soon with the next generation of therapeutic drugs. Actually, genetics plays a major role in Alzheimer's. It's one of the ones where our predictors are most powerful. So if it were the case that there were things individuals can do, and there are some lifestyle changes that they can do to impact their risk currently, but as we get closer to having therapies which will help with prevention, I think that is a real opportunity.
Looking at all the common diseases that we've talked about, what is it that you would like to happen next? What would be, if you had a magic wand, what would you like the next step to be?
I think the obvious next step is to roll out these technologies in cardiovascular prevention, so prevention of heart disease and strokes. Those are situations where doctors already estimate risk. Genetics is an important risk factor. It's individually at least as important as the ones we currently use, like blood pressure and cholesterol level. But it's not being used currently. We've shown that it can be measured, it can fit in with workflows, and it makes a difference. So that's a very natural place to start. Another area where I think genetics has an important role to play that is underappreciated is in obesity, where we and others have shown that genetics is a major driver of the likelihood of a person becoming obese and then the challenges they have in losing weight when they do. A recent study by others showed that as an individual, risk factor is probably twice as important as lifestyle factors. Actually, we've redone the analyses with a more powerful genetic tool, and maybe it's 3 or 4 times more powerful. And just that understanding that genetics is a big part of the story around obesity, the fact that for some people, it's really not about lack of willpower or they're not trying hard enough. Their brain is wired differently, and the way it reacts to signals from their stomach when they've eaten and their metabolism, those things are just different in some people that mean it's much harder for them to maintain a stable weight. But we need to understand that. We can identify those people, we can give them more support. And that's exactly what we should be doing.
Peter, thank you very much. It's been absolutely fascinating. This first episode of the Genomics Revolution, it's been wonderful to hear it in action. Thank you for joining us.
It's been a great pleasure. Thank you very much, Sian.
And if you've enjoyed this episode, please join us again. Sign up and subscribe, and you'll find our contact information in the description below.